Welcome to our channel Mootechie, Measurement of Bacterial Growth. This presentation provides a clean modern overview of the direct and indirect methods used to measure bacterial growth, along with their pros, cons, and applications. Why measure bacterial growth? There are several key reasons. First, to monitor population dynamics and growth phases. This helps us understand how a bacterial community changes over time. Second, to optimize bioprocess or fermentation yield and timing. Knowing exactly when bacteria are most active allows for better control in industrial settings. Third, to guide therapy, specifically antibiotic susceptibility and dosing. Accurate growth measurement tells us if a treatment is working. Fourth, to ensure safety in food, water, and pharmaceutical quality control compliance. We need to know if harmful bacteria are present. And finally, to track ecology in environmental and microbiome studies, helping us understand the role of bacteria in various ecosystems. Now, let's look at the direct methods for counting cells. Direct methods, cell counting, part 1. The first method is the microscopic count using a hemocytometer. This gives a total cell count, is fast but often requires staining to see the cells clearly. Next is the plate count or CFU method. This counts only viable cells through serial dilution and either spread or pore plating, resulting in visible colony-forming units. The third method is MPN or most probable number. This is a statistical estimation for counting viable cells in liquid samples. Finally, epifluorescence microscopy uses viability stains to distinguish live from dead cells under a fluorescent light. Looking at the diagrams, we see a visual comparison. The hemocytometer allows for direct microscopic observation. The plate count shows serial dilution leading to isolated colonies on a plate. The MPN method is represented by test tubes with different dilution levels estimating a statistical count, and epifluorescence shows the use of fluorescent dyes to determine cell viability. Moving on to direct methods of microbial enumeration, concentration and instrumentation, part 2. Method 1 is membrane filtration. This concentrates low biomass samples and colonies are then counted directly on the filter. Method 2 is electronic counting, like with a Coulter counter. It provides size-based detection for cell counts. Method 3 is flow cytometry. This allows for rapid single-cell enumeration and can be combined with viability dyes. Method 4 is qPCR or dPCR. These techniques quantify gene copy number, offering extremely high sensitivity for detection. These instrumental methods are powerful because they automate counting, handle large sample volumes, or detect cells based on specific properties like size, light scattering, or genetic material. Now, we transition to indirect methods, optical and metabolic signals, part 1. These methods measure a proxy for growth, not the cells themselves. The first is turbidity, measured as OD600. The optical density of a culture correlates directly with its biomass. Second is resazurin or MTT deduction. These assays measure metabolic activity as a proxy for living cells by tracking electron transfer. Third is pH or acid production. In fermentations, bacterial activity shifts the pH of the media, which can be measured. Fourth is gas evolution, like CO2 or H2. The production of these gases changes pressure or volume, serving as an indicator of growth. The simple graphic shows an upward trend for an OD signal and a separate indicator for metabolic activity, highlighting that these are two different types of indirect measurements we can track. Continuing with indirect methods of microbial growth measurement, mass and composition, part 2. The first method is dry weight. Here, culture is filtered, dried to a constant mass and weight to get total biomass. Second are protein or nitrogen assays like Bradford, Biorette or the Kelgel method. These measure cellular components that increase as cells grow. Third is ATP bioluminescence. This uses a luciferase enzyme to produce light in the presence of ATP, a direct signal of metabolic activity and viable biomass. Fourth is conductivity or Brix measurement. These track changes in the ionic strength or sugar concentration of the media, which often correlate with microbial growth. These methods are useful when you need to measure total biomass or specific cellular components, especially when direct counting is difficult. It's important to understand the advantages and limitations of direct versus indirect measurement. Direct methods like plate counts or flow cytometry offer accurate absolute or viable counts with high specificity. However, they tend to be slower, more labor-intensive, and have a longer time to result. On the other hand, indirect methods like turbidity or ATP bioluminescence are rapid, high-throughput proxies for growth. They are great for automation and getting quick trends. Their limitation is that they require calibration against direct methods, and the readings are inferential and can depend on the sample matrix. The slight contrast plate count and flow cytometry for direct methods against a spectrophotometer for OD600 and a luminescence readout for ATP, visually representing the different approaches. So, where are these growth measurements applied? The key applications are diverse. In the clinical field, for bacteremia or UTI workups, tracking minimum inhibitory concentration and guiding therapy. For food and water safety, it's used in HACCP plans, shelf life testing, and coliform monitoring. In bioprocess industries, it's critical for scale-up, feed control, and determining the optimal harvest time. In environmental science, for monitoring wastewater, soil, and biofilms. And in research and education, for constructing growth curves and phenotyping mutant strains. The icons highlight these areas: a lab flask for clinical diagnostics, a leaf and water droplet for food and water safety, a bioreactor for bioprocess, a plant for environmental studies, and a growth curve graph for research. Here is a summary and the key takeaways from our session. Choose direct methods like CFU or flow cytometry when you need absolute or viable cell counts. Choose indirect methods like OD600 or ATP assays when you need rapid trends and automation. It's crucial to calibrate your indirect proxies to your specific bacterial strain, growth medium, and instrument path length. Always standardize your standard operating procedures, controls, and quality checks to ensure reproducible growth curves. For the best balance of speed, accuracy, and cost, consider combining both direct and indirect methods in your workflow. Thank you. I hope this exploration of how we measure the unseen world of bacteria has been insightful. Understanding these tools empowers us to ensure health, drive innovation, and protect our environment. Thank you for learning with us.